Hi, everyone. Today we're going to finish up with our discussion about the different components of the cells, the different organelles. In our last video, we talked about ribosomes, endoplasmic reticulum, lysosomes, and vacuoles. Today we're going to focus mostly on mitochondria and chloroplasts. And remember, as you go through these, I want you to pay attention to how the structure is related to its function. So let's take a look at the mitochondria. Now, the structure of the mitochondria, it has two membranes. It has a double membrane. The outside one is very smooth, and the inside one, which is sometimes called the cristae, has multiple folds, and those folds increase the surface area available inside the mitochondria. And that inner membrane is where the electron transport chain and where it is located and where ATP synthesis occurs. So the more surface area you have on this membrane, the more ATP you can produce. So it kind of makes a lot of sense that the inner membrane has many folds in it. Interesting thing about the mitochondria is that the mitochondria has its own mitochondrial DNA separate from the chromosomes in the cell. And the mitochondria has its own ribosomes, separate from those inside the cell. And those ribosomes are very similar to the ribosomes found in some types of bacteria. So the fact that mitochondria have their own DNA and their own ribosomes are a couple pieces of evidence that mitochondria may have been their own independent living bacteria at some point in evolutionary time. And that's called the endosymbiont hypothesis, which won Dr. Lynn Margulis the Nobel Prize. And what Dr. Margulis's hypothesis was that the mitochondria and the chloroplast, which we'll get to in a minute, both were independently living bacterial cells. Mitochondria were originally bacteria that were very good at energy production. They got swallowed up by bigger cells and formed a symbiotic relationship where these mitochondria provided energy to the bigger cells, and the bigger cells provided maybe protection or food to the bacteria that used to be the mitochondria. As with many scientific theories, Dr. Margulis's endosymbiont hypothesis was widely ridiculed when she first proposed it in the 1960s. And then I think about 15 years later, maybe it didn't even take that long, she actually won the Nobel Prize for it. Now, the chloroplast, like the mitochondria, has different membranes. The outer membrane is smooth again. And then inside, instead of having just one inner membrane, there are multiple inner membranes, like little stacks of pancakes, okay? Again, the chloroplast has its own chloroplast DNA and its own ribosomes that are similar to ribosomes that are found in bacteria. More evidence for the endosymbiont hypothesis that chloroplasts used to be independently living bacteria. Chloroplasts, of course, function in photosynthesis in both algae and plants. And now the thylakoids are what we call these little membranous stacks inside the cell, the stacks of the grana. Okay, and it's on those thylakoid membranes where the chlorophyll molecules are located, and also the electron transport chains of the different photosystems. The light-dependent reactions of photosynthesis, sometimes called the light reactions, you can think of it as maybe being the first half of photosynthesis, occur on the thylakoids inside the chloroplast. Now, I tell students that the thylakoids look like a stack of pancakes. What do a lot of people like to put on pancakes? Syrup. Except there's not syrup. There's a syrupy liquid inside the chloroplast, but it's called stroma. So stroma refers to any fluid inside the chloroplast, but outside of the thylakoid. And the stroma is where the Calvin cycle, aka carbon fixation, also known as the light independent reactions of photosynthesis, occur in the stroma. And some people refer to that as the second half of photosynthesis, the light independent reactions, carbon fixation, the Calvin cycle. And that's it for today. I hope that makes sense, and I hope you guys have a great day. Bye.